Can you see now that it's live on YouTube?
I know
So
I'm not even on. I'm not on YouTube.
no. Okay, can you not see it on Zoom? I mean, at the top left, it's. I don't know if it's just me who can see it. I'm not sure. Yeah, maybe
What
that's.
what what I can see is you are screen sharing live.
Yeah. Anyway. So, sir, we can start immediately. I'm just gonna start
Okay, so
recording. I will start record. Can you go to the very first page, please, where it says characteristics
yeah,
of
this
living things? Yeah.
yeah yeah. Can you see it?
All right. All right. So I'm gonna start recording now immediately. Yeah. You can start
Okay.
now, please.
Yeah. All right. So Tammy, can you see my screen? Tammy, can you see my screen? I'm not getting any feedback.
Yeah. There's maybe time lag. Let me just type in the question.
Yes, sir.
Oh, okay. The feedback is not that clear. All right. So I want to run through characteristics of living things with you. And in this particular lesson, we'll be looking at different characteristics, the little things that we should note concerning each characteristic. And we'll also be trying to answer a few questions, a few JAMB questions that you may likely come across when you take your JAMB exam. So first of all, we're going to look at movement. Movement is regarded as a change in position. And that simply means that living things have the ability to change their position from point A to point B. Now, the way this happens in plants and animals is quite unique to both groups of organisms. For instance, in plants, their movements are usually what we describe as apical, and it is related to growth movements. For instance, plants tend to grow towards sunlight, and the fact that there is a change in position means that we can describe that as movement. Animals, on the other hand, exhibit what we refer to as locomotion, which means that they can move their entire bodies from point A to point B. Of course, if we are talking about things like algae, some of them actually can exhibit locomotion, but we're not going to discuss that under this particular topic. So we look at the next one, which is growth. Growth is referred to as an irreversible increase. The parameters would be size, mass, and number. So if you have an irreversible increase in size, you would call that growth. If it's an irreversible increase in mass, you would equally call that growth. Or if you're dealing with cells, whether you're dealing with microorganisms or you're dealing with multicellular organisms, if you have an irreversible increase in the number of cells, then you would describe that equally as growth. Another synonym that you can use for the word irreversible will be the word permanent. Then we move on to reproduction. Reproduction has to do with the ability of organisms to give rise to offspring. Okay, the whole idea is to replace themselves and ensure continuity of life. So let's move on to number 4. Number 4, we have respiration. And respiration has to do with the breakdown of food. I wrote glucose here because most of us are familiar with glucose, but it does not necessarily have to be glucose. In any case, respiration has to do with the breakdown of glucose in order to release energy. So our key words are breaking down of food and then releasing energy. Of course, we know that there are 2 types of respiration. We have aerobic respiration and we have anaerobic respiration. Aerobic would occur in the presence of oxygen, and anaerobic would occur in the absence of oxygen. So whether it's aerobic or anaerobic, the goal of respiration is to ensure that food is broken down and energy is released. The energy can be used for a variety of life processes. So let's look at number 5, irritability or sensitivity. Irritability has to do with the response to stimulus. And a stimulus is described as an environmental change. Now, your stimulus could be an environmental change. It could be an internal environmental change, or it could be an external environmental change. Either way, you can identify a stimulus within the context of, let's say, temperature. Change in temperature definitely would be a stimulus. Change in pressure would equally be a stimulus. Next, I would move on to number 6, which is nutrition. Nutrition has to do with the intake of food. And we take food for growth, energy, and a variety of things that occur within us. Number 7 would be excretion. And as far as excretion is concerned, we are talking about the removal of metabolic waste. Now, when we're talking about excretion, the word metabolic is very, very important. Because if we are not using that word metabolic, then we are not talking about excretion. We most likely will be talking about egestion. So there is a difference between excretion and egestion. So what is the difference between metabolic and non-metabolic waste? When we use the word metabolic waste, it means that you have taken in food, the food has been digested, okay, compounds have been made. Now, part of those products that have been made from the digested food, some of them are going to be utilized by the cells, others will be regarded as being in excess of requirements. And so these ones that are in excess of requirements would have to be released from the body. So when we use the term metabolic, we're talking about the food that has been broken down and used to make certain products, some of which have been used and some of which are not required anymore. So they have to be excreted from the body. On the other hand, when we use the term egestion, egestion simply means that you are removing solid, undigested waste. So while your feces are regarded as waste of digestion, examples of your metabolic waste would be things like sweat, carbon dioxide, urea. All these things are produced by your body, and they are not required, so they have to be excreted from your body. Then we move on to number 8, which is adaptation. As adaptation is concerned, as far as adaptation is concerned, we are dealing with the properties, the characteristics, the traits, the features. Okay, you can use the word characteristics, you can use the word features that help organisms to survive better in a given environment. We could equally rephrase this as the features that improve the chances of survival of an organism in a given environment. Next, we move on to competition. And as far as competition is concerned, we're talking about organisms struggling for limited resources. The fact that resources are not always abundant within the ecosystem or within the environment, it means that organisms will have to struggle for these limited resources. And some of the examples of the resources that we are talking about are light, space, food, water, even mates. And then finally, we have death. Death has to do with a cessation of life functions, which means the life functions of movement, respiration, growth, excretion, reproduction, all of them will cease to happen. So any questions before I move on?
Let me just ask her in the chat box, and I might have some questions myself because I'm a student. So, yes.
Okay.
Yeah, she has a question, so she's gonna just type it in the chat box.
I hope I can see this chat box here. Yes, I said growth is irreversible. Yeah, so yeah, I'm still waiting for the question.
I think she's trying to say that, yeah, she's just like asking, is that what you said, that growth is irreversible?
Yes. Yeah. Yes, it's irreversible. Because if, for instance, let me go back to that. Yes. So if, for instance, now, today, let's say I'm weighing about 90 kg today. And yesterday, I was weighing about 80 kg. And then tomorrow, I come back and then you weigh me and I'm now weighing 85 kg. We cannot say that I am growing. Okay? Because obviously, what is responsible for the increase is not what we call biomass. Okay, it's not what we call biomass or what have you, right? It may just be that I'm eating more and more by the day. Maybe between day 1 and day 2, I overate and I did not go to the toilet, and so I was weighing more. That does not mean that I have grown. For you to say that I have grown, it means that the number of cells in my body must have increased irreversibly. Okay, but people that lose weight, yes. So this is the thing. You have to look at it longitudinally. When you were 3 years, there was a certain weight that you had. When you were 6 years, there's a certain weight that you had. There is no way you will go back, all things being equal, there is no way you would go back to the weight that you had in when you were 3 years. So that is what we mean by growth. The mass that you have accumulated, you cannot, if it's an indicator of growth, okay, you cannot reverse it to that mass that you had when you were 3 years. Okay, I think so we have our mind map here. I hope you can see. You can see characteristics of living things, and then you can see a summary of everything that I just explained, from growth to death, from movement to irritability. So at a glance, you can easily remember everything that I've talked about today. So to just help us, let's see. Okay, before I even go into the question, I think you also have a question.
Myself, sir?
Yes. Yes.
Yeah, my question is there any mnemonic, or like how can I recall these characteristics of living things?
Yes, there are 2 mnemonic, but you would have to add adaptation, competition, and death to it. The one that most people are used to is Mr. Niger D. So it's not too long ago that they added competition and adaptation. So if you like, you can call it Mr. Niger Dark. So
all right.
if you use Mr. Niger Dark, then you should be able to remember. Or if you like, you can reverse this and call this Mr. Niger Card. Anyone?
Thank you, sir.
Yeah. So let me look at the questions that we have here. Suggested revision questions. All living organisms. Dash. Is it A, photosynthesize? Is it B, have plastids? Is it C, transpire? Is it D, conjugate? Is it E, feeding? So if we use a mnemonic, Mr. Niger Duck, or card, which one tallies with the letters that we have there in the mnemonic? Is it photosynthesis? Is it plastic? Is it transpiration? Is it conjugation? Is it feeding? So, Tammy, can you suggest what the answer is? Okay. So, according to Tammy, it is feeding. This is a very, very simple question. Okay. Then let me move on to the other one. Irritability is also called, is it movement sensitivity, respiration, or development? Tammy? Okay, so she said it's sensitivity. And that is that for characteristics of living things. So we will try to see if we can also take cell structure and functions of cell components. Yeah, so of course, we know that the cell is the basic unit of life, or we could basically describe it as the basic structural and functional unit of life. And in analyzing the cell, we find out that one, we do have quite a lot of unicellular organisms, which are organisms that exist as single cells, right? They can function on their own without depending on any other cell. And then even when we talk about multicellular organisms, we do find out that even though they work together, within each cell, we do have organelles that perform unique functions. Now, if we look at unicellular organisms, our examples will include amoeba, chlamydomonas, euglena, paramecium, and so on and so on, and so on and so forth. So for multicellular organisms, of course, I've already reiterated the fact that they depend on each other. They consist of cells that depend on each other, and each cell consists of organelles that have unique functions. So if we look at unicellular organisms, for instance, maybe I should just take the organelles before looking at the unicellular organisms. Anyways, so for a typical mammalian cell, they have two major parts. And one is what we call the protoplasm. The second is what we call the plasma membrane. So for the protoplasm, we describe this as the living part of the cell. Okay. For the plasma membrane, this is the cell boundary. And we'll talk about the uniqueness of the plasma membrane in a while. So for the protoplasm, we've already itemized that it contains the nucleus and the cytoplasm. Within the cytoplasm, we have a vast array of organelles, which we'll still talk about much later. Well, first of all, I would like to talk about the plasma membrane. So the plasma membrane in itself, like I said, is a boundary around the cell that regulates what goes into the cell and what leaves the cell. And its property is linked to the fact that it contains what we call phospholipids. Now, if you cannot remember the word phospholipids, at least you remember the fact that your plasma membrane contains lipids. It also contains proteins. So just to add a little bit of information, the proteins function as channels through which certain substances enter or leave the cell. So let's look at the organelles within the cell. It is important for us to be able to identify each organelle that we have in the cell. But we'll just, even though this particular diagram shows quite a lot of organelles, we'll look at the most important ones, starting from the nucleus in the center. The nucleus is where you have your chromosome. And keep in mind that your chromosome is made up of DNA. Your DNA contains genes. So when we say that the nucleus directs the cell activities, what we are actually saying is that it contains genetic material that directs the cell activities. If you look around the nucleus, you would see a membrane around it. This membrane has gaps. Now, this membrane around the nucleus is what we refer to as the nuclear membrane. And the fact that it has gaps in between it means that materials can enter the nucleus through those gaps and leave the nucleus through those same gaps. Within this nucleus, we can see a dark staining region, which we refer to as the nucleolus. And the function of the nucleolus, in actual fact, is to make ribosomes, which will take us to another part of the nucleus. Okay, so these are ribosomes, the dot-like structures that we see within the cell. The function of these ribosomes is to synthesize proteins. Okay, now when these proteins are being synthesized, it is the job of this system, this interconnected membrane-bound structure that we see here. Now, this interconnected membrane-bound structure, we refer to it as the rough endoplasmic reticulum, RER for short. It also has another, so let me just circle this so that you know what I'm talking about. Okay, so it also has another system of interconnected passages, membrane-bound passages, which we refer to as the smooth endoplasmic reticulum. The rough endoplasmic reticulum is associated with ribosomes. So after the ribosomes have synthesized their proteins, it is the job of the rough endoplasmic reticulum to transport these proteins. Now, the question now is, where would they be transporting the proteins? They would be transporting the proteins to the Golgi apparatus. So which means that the function of the rough endoplasmic reticulum is connected to the function of the Golgi apparatus. And what the Golgi apparatus does is to modify the proteins. But in JAMB and WAEC syllabus, we are not satisfied with just saying modify. We say synthesis of enzymes and hormones. Now, the enzymes that we're talking about, they are made up of proteins. So it makes sense for us to talk about the fact that the proteins coming from the rough endoplasmic reticulum will be modified by the Golgi apparatus. And part of the modification process will lead to the production of enzymes, which, like I said earlier, are proteins. We also have hormones that are protein-based. So the synthesis and transport of these enzymes and hormones would be the function of the Golgi apparatus. If you look closely at the Golgi apparatus, you would also see that there are vesicles being formed. Now, these vesicles contain these enzymes and hormones. And the issue here is that depending on where they are required, the vesicles will either fuse with the cell membrane to release them or transport them elsewhere within the cell where they are required. Now, one significant role of the Golgi apparatus is the production of this structure here, which we call the lysosome. And the lysosome, from our observation, is a spherical structure that contains hydrolytic enzymes. This, you can stop me anytime while I'm explaining so that I can give you a little bit more clarification concerning what I'm talking about.
So I have a question, please.
Yes.
My question is, as a student, do I need to know how to draw and label this structure?
Well, for JAMB, you don't need to know how to draw the structure, but you need to know how to label, how to identify, and you need to know the functions of these organelles.
Thank you. So I need to know all these like the different organs, nuclear organelles, all of them.
Well, yes, this is the thing. You will notice that I didn't talk about everything. For instance, I didn't talk about this. I didn't talk about this. And I didn't talk about this. So it's not everything that you need to know.
Okay.
But just, yes, there are certain organelles that are specified in the syllabus that you need to know. And even if you don't know everything, you should know just enough to help you understand or be able to figure out the answer, since it's a multiple choice based exam. Okay, so Tammy, do you have any questions before I move on?
No, sir.
Okay. So the lysosome, like I said, is a product of the Golgi apparatus. And we already know that the Golgi apparatus synthesizes and transports enzymes and hormones. So when I say that the lysosome contains hydrolytic enzymes, it makes sense. The question now is, what would be the function of these enzymes? So these enzymes are implicated in phagocytosis and they are also implicated in breaking down worn out organelles. Okay. So I'm going to talk about two more organelles, one being the mitochondria. The mitochondria is for ATP production, particularly in aerobic respiration. Now remember that I said we have aerobic and anaerobic respiration. Mitochondria is not interested in anaerobic respiration. It is only interested in aerobic respiration. So it only works when you have oxygen present. And then finally, this is a word that most students at this level will not be familiar with. So let me write the words that they will be familiar with. It is called centriole. All right. So centriole actually produces what we call spindle fibers. And these spindle fibers, we talk about spindle fibers during nuclear division, specifically mitosis. I would still come back to this issue of spindle fibers when I'm talking about the differences between plant and animal cells. But just to have a recap, we have a plasma membrane that regulates what goes in and out of the cell. We have a nucleus that contains chromosomes or DNA that directs the cell. We have a mitochondria that is involved in energy production. We have a rough endoplasmic reticulum that folds and transports proteins made by the ribosomes. We have a smooth endoplasmic reticulum, which is responsible for synthesis and transport of lipids and steroids. We have a Golgi apparatus that synthesizes and transports enzymes and hormones. So, and then finally also we have a cytoplasm within which all these organelles are suspended. Without this cytoplasm, all these organelles will just clump in one particular place within the cell. Now, the organelles that I've described are present also in the plant cell with a few exceptions. For instance, in the plant cell, we have a cell wall. And the function of this cell wall is actually to prevent the plant cell from bursting when it is experiencing high sugar pressure. Now, you may be wondering what sugar pressure is. So the plant cell experiences sugar pressure when it continues to take in water. The pressure exerted by the water on the cell wall is what we refer to as sugar pressure. Now, if it was an animal cell, as it continues to accumulate water, eventually it is going to burst because the cell membrane is not as tough as your cellulose cell wall. So what your cellulose cell wall would do is to provide a counter pressure so that the plant cell only becomes turgid, it does not burst under high sugar pressure or when it's experiencing osmotic pressure. Then we've looked at the cell wall. So what else do we have in the plant cell that we do not have in the animal cell? We have the chloroplast. The function of the chloroplast is to carry out photosynthesis. All right. Next would be this vacuole. Within the plant cell, we have a large vacuole. And because this vacuole is so large, it pushes the nucleus to the periphery, to the edge of the plant cell. Now, in this particular diagram, the illustrator has not particularly represented the size of the vacuole very well, because usually the size of the vacuole should cover almost the entire length, or should I say almost the entire volume of the plant cell, thereby pushing this nucleus to the edge. So three things that we have learned here is the fact that the plant cell has a cell wall, a structure that we do not find in the animal cell. It has a large vacuole. Of course, we will talk about the function of the vacuole later. And then it equally has chloroplasts, which are responsible for photosynthesis. Okay? So that summarizes. So what I will be doing right now is basically recapping what I've talked about and then adding a little bit of information here and there. For the plasma membrane, we've already talked about the function. Then for the nucleus, we've talked about the fact that it controls the cell activities, houses chromosomes, contains the nucleolus. Please tell me, you can call my attention at any point in time. Then the cytoplasm, we refer to that as a jelly-like solution of fluid that suspends other organelles. And like I said when I was explaining, without the cytoplasm, all the organelles in the cell will just clump in one particular place, which doesn't particularly make any sense. Then, for the chloroplast, we established the fact that the chloroplast, the function of the chloroplast is to carry out photosynthesis. What we did not say is that the chloroplast contains what we call grana. And these grana contain chlorophyll. Now, the chlorophyll is the pigment that actually traps sunlight and initiates the process of photosynthesis. For the mitochondria, we've established that the function is to produce energy in form of ATP. But when we're talking about producing energy in form of ATP, we are actually talking about the process of aerobic respiration, not anaerobic respiration. Now, one unique thing about the mitochondria is the fact that it actually has two membranes. The outer membrane is smooth, continuous, whereas the inner membrane is folded. And then we'll discuss why this is the case when we are talking about adaptation. So we look at the cell wall. We talked about the protective function of the cell wall, protecting the plant cell from bursting under high sugar pressure or osmotic pressure. Some students say that the cell wall is to maintain the shape of the cell. Not quite. The cell wall is not to maintain the shape, it's basically to prevent it, it's protective and to prevent it from bursting. Then we looked at the vacuoles. Now, one unique thing with the vacuole is the fact that in plant cells, the vacuoles have a variety of functions, majorly storage, and they store waste products as well as pigments within the plant cell. If you find a vacuole in an animal cell, it is usually small, temporary, and usually it is either what we call a contractile vacuole or a phagocytic vacuole. So when we talk about contractile vacuole in animal cell, this is for excreting excess water. When we talk about a phagocytic vacuole, this is a vacuole that encloses bacteria or other unicellular organisms with a view to further digesting them. Then we have ribosomes. Like I said, ribosomes are for protein synthesis. All right. I talked about the endoplasmic reticulum. We have the smooth and the rough. The rough is the one that is connected to ribosomes, and so it is the one that is responsible for folding and transport of proteins. And then the smooth endoplasmic reticulum, like I said, is for synthesis and transport of lipids and steroids. Golgi apparatus has to do with transporting enzymes and hormones. Lysosomes have to do with phagocytosis as well as destruction of worn-out organelles. Centrioles have to do with the production of what we call spindle fibers. And one other thing you should really take note of, apart from the fact that they produce spindle fibers, is the fact that you find them near the nucleus of animal cells. This tells you that you don't find centrioles in plant cells. You only find them in animal cells. And to locate them, you need to look near the nucleus. All right, so before I move into the similarities between plants and animal cells, Tammy, do you have any questions?
No, sir.
Okay, so because I'm not sure we'll be able to, we only have 4 minutes remaining, but in a lot of ways, I have talked about the differences between plants and animal cells. For instance, that your plant cell contains chloroplast, the animal cell does not have chloroplast. The plant cell, additionally, we can talk about this point here. The shape is polygonal, meaning it has many sides. It could be hexagonal, okay? But for the animal cells, they are usually roughly spherical. And for the plant cells have a cellulose-based cell wall, while the animal cell does not have a cell wall at all. The plant cell has large vacuoles. The animal cells are small, if at all. The plant cell will store its lipid as oil, which is why you have vegetable oil, and while the animal cells will store its lipid as fat. which is why you have cholesterol from your chickens and your cows and the rest. Now, the nucleus in the plant cell, like I said earlier, is pushed to the edge, whereas the nucleus in the animal cell is roughly central. Centrioles are absent within the plant cell, and within the animal cell, they are present. For carbohydrates and the forms in which they are stored, we find out that carbohydrates are stored in form of starch in plant cell, and then they are stored in form of glycogen in animal cell. So this is our mind map, where we have the structures that we find in both plant cell and animal cell on the right, the structures that we find only in plant cells on the left. I believe we can answer a few questions. So, Tammy, can you answer the first question? Which of the following structures differentiates an animal cell from a plant cell? You can type. Okay, so Temi said cell membrane. So this is what we're going to do. I will take you back to the mind map, and then you'll answer the question again. Have you taken your time to look at the mind map? Okay. I'm going to assume you've taken your time to look at the mind map. So you can attempt the question again. Okay. All right. So this is the thing. The question is saying, which of the following structures differentiates an animal cell from a plant cell? Okay. Now, your ribosome is present in both animal cell and plant cell. Lysosomes will be present in both animal cell and plant cell. Cell membrane will be present in both animal cell and plant cell. Mitochondrion will be present in both animal cell and plant cell, but chloroplast is only found in plant cell. So I think we can end here for today. Sir?
All right. Thank you very much for having the time. It's a pleasure. Thank you, Temi, for attending this class.
Yes, sir.
Yeah, so thank you very much for attending this class. Let me just stop the recording first. Yeah, I'm gonna stop the recording. Yeah. Our next class, biology class will be next Saturday. We have English class on Thursday, and physics class will start on Tuesday, hopefully. So I will come. Are you taking physics?
Yes, sir.
All right. So yes, thank you very much, sir. I'm just going to also end the live streaming. We've been successful this time around.
Yeah. I hope so.
Yeah, all right. So thank you very much for attending this session. Until
Okay, no
next week.
problem.
All right, goodbye.
All right.
Goodbye.